I would first deeply thank my friend Derek to inviting me as a keynote speaker. And I'm very pleased and honored to introduce our recent research finding related to begomovirus resistance. And I hope our knowledge could somehow help the farmers and contribute to the development of pepper production. So briefly, I would like to introduce my personal journey related to peppers. So since my undergraduate years, I've been working with peppers. Mostly in previous studies, we use Capsicum chinensis. And I work for several different pepper biology, such as temperature response, capsaicinoid biosynthesis, and volatile biosynthesis. But today, I would like to introduce about the begomovirus resistance, which I have been we have been working most extensively during past 15 years. So, as you may know, capsicum is a very important horticulture crops, and in the world, about 50 million tons are produced, and the biggest producers are China, India, Turkey, Mexico, and Indonesia. They produce more or less 75% of all production. And many of these countries, not only these, but suffer a problem with begomoviruses. So initially, when I started this research, I didn't even know begomovirus or Gemini virus. And this was taken in 2010 in Indonesia. I had a chance to visit, and this is typical open field for pepper farmers. And if you get close to the plant, you can easily see the disease symptom. It's a viral disease. I didn't know what the pathogen was, but I could understand that no fruits can be harvested, and this will critically affect the farmer's income. And because there is no pesticide for viruses, farmers cannot earn money. That is a problem. And I thought at least it should be a huge problem in Indonesia. And went back to Japan and searched for publication and found that this is actually a global issue. And there was not so much progress when I started this research. So as a young scientist, 15 years ago, I'm already mid-age, but I chose this as a good topic for myself. So now, Gemini virus family contains 15 genera, and begomovirus genus is the biggest one, which contains more or less 460 species. And they are transmitted by a whitefly, a small insect vector. So what we do is to treat insecticide and put insect-proof mesh around the greenhouses. But most of the country cultivate pepper in the open field, so this is not enough. We need a virus-resistant variety. So the situation in pepper, we have several hotspots for the begomovirus disease. One is Southeast Asia, also in Thailand, I guess, and South Asia, West Africa, Central, and South America. There are different species, different predominant begomovirus species in each area, but mostly they induce similar symptoms, leaf yellowing and coloring symptoms. And for example, in India, they have so many DNA component, which is a virus species, and also DNA-B satellites are combined in a company. So it's almost impossible to understand the virus diversity, but somehow we have to control the disease. So for this purpose, I want to support the breeding. So initially, we did field screening in Indonesia. But we soon faced difficulties because in the field, of course, begomovirus infects, but as a rhinovirus, as a fungi or bacteria, and there's heat stress, water stress, everything is mixed up. So it was very difficult for us to select the good begomovirus resistant source. And in the meantime, there were several publications, mainly from India, also Derek published a good paper. Many people also did a field screening. So to find my originality, I chose to go back to the laboratory condition. So in our research, we use controlled climate condition in the growth room using a small seedling and also pure virus isolate. This is also important. And to avoid using insect vector, we prepare the infectious clone, which we use agrobacteria for inoculating begomovirus to pepper. So these are all for accurate phenotyping, because we need phenotyping always, and if we combine the genotype data, we can do a good work. So our research can be divided to 5 different stages. In the stage 1, I was more like a biologist, collect sample in the field, do the diagnosis, and identify the predominant virus species. And in the stage 2, we developed the inoculation method using infectious clone. And next, we screened from germplasm, and we also did identification of resistant genes. And in the end, we did a gene pyramiding to enhance the resistance. So in case of our begomovirus, it is a bipartite begomovirus, which have DNA A and DNA B. Both are circular single-stranded DNA. And initially, we clone full-length and partial-length sequence in the single plasmid for DNA A, and for DNA B, we clone in the different plasmid and transform the agrobacteria and do the inoculation. This works well for such as Nicotiana benzamiana or tomato, but if you do the same in peppers, it didn't work. So we cloned partial repeats of DNA A and B together in a single plasmid like panel D, and like panel C, we observed a similar symptom as a field, which is a panel D. So using this method, we screened about 900 pepper accessions, including all the domesticated species. And we selected 2 promising material, which is BAPEP5 and another is PG11. BAPEP5 is a local accession from Indonesia, and PG11 is we introgressed resistance from Capsicum pubescens to Capsicum annuum, which is very difficult. And we cloned resistant genes from these 2 accessions. So first, I would like to start from BAPEP5. So we use 2 virus species, PEP5CIB, PEP5CAB, both are from Indonesia. And for example, when we inoculate to BAPEP5, it's almost symptomless, but susceptible BAPEP4 is showing yellowing, and also the F1. So this resistance is recessive resistance. And when we check the viral DNA accumulation, the red bar is BAPEP5. Accumulation is significantly restricted. So we can say this line has a resistance against begomovirus. So next, we prepare the F2 population, about 500 individual we inoculated, and we use only 300, more or less 50 plants which were positive for virus infection, because we don't want to use an inoculation escape for our genetic analysis. And 28 were resistant, 380 was susceptible. We also checked the viral DNA accumulation data, and using these data as a phenotype data, we did high throughput sequence to do the mapping. So as you can see in the lower graph, there was a single peak on chromosome 5. So because this was the first gene in pepper, we named PEPY1 following the famous TY gene in tomato. And in this case, we were very lucky. We just developed additional DNA markers and narrowed the candidate region. In the end, 34 kb, which contains just 1 single gene. And when we checked the annotation data, it found out it encodes a protein called pelota. This is famous because tomato TY5 also encodes pelota. We didn't expect, but in the end it was. So anyway, we compared the genomic sequence and also the mRNA sequence, and there was just a single SNP on the 9th intron, A to G. And because of this SNP, 9th intron was not spliced, but rather translated to amino acid and included in the protein. So by this, we have additional publication, but the function as a payload has significantly decreased, not disappeared, but decreased. By this, Pepper acquired begomovirus resistance. So, and to prove that pelota mutation is actually conferring the begomovirus resistance, we did virus-induced gene silencing. In this case, we use pepper susceptible to begomovirus. So in the middle, when we inoculate begomovirus with Big's virus, plant is still susceptible, but when we silence the pelota expression and inoculate begomovirus, it becomes resistant. So it means pelota is actually the PEPY1 itself. And for breeding purpose, we develop a good CAPS marker. On the causal SNP, we can easily distinguish the resistant and the susceptible genotype, and the phenotype 100% match with the genotype. I think many people are now using this marker for breeding. And for PG11, we did similar approach. We first evaluate two parents, susceptible SCM334 and resistant PG11, and viral DNA accumulation was significantly restricted. And in this case, segregation ratio was very strange because we introgressed resistant from Capsicum pubescens. But among 270 plants, one plant was susceptible, 260 was resistant. So we can still do the mapping and detected a peak on chromosome 7, which is different from the previous PEPY1 on chromosome 5. And then in this case, we developed a F3 population to narrow down. And in the end, we had a candidate lesion which contains 70 genes. And then we checked the annotation data. And again, gene was same with tomato. There is a famous gene TY1, TY33A in tomato, which encode RNA-dependent RNA polymerase. Also same in this gene PEPY2. So when we compare the sequence between susceptible SCM between PG11, in the susceptible parent, there was at least 3 premature stop codon to induce loss of function as a RDR. And when we draw a phylogenetic tree, pepper RDR graded very close with tomato RDR in the same gamma grade RDR. And interestingly, because there are several whole genome sequences in the public database, we checked the RDR sequence. Most of them are Capsicum annuum, some are from Capsicum chinense and bacatam. All of them had a loss of function mutation. So we can say only some accession of Capsicum pubescens has resistant allele. And we have successfully introgressed to Capsicum annuum. And we developed a DNA marker in the gene, so we can distinguish the genotype and predict the phenotype perfectly. And in the end, we did the Bigs again. In this case, we use the PG11. So when we co-inoculate begomovirus with Bigs virus, PG11 became susceptible. So we conclude that PEPY2 is encoding RDR. So in case of PEPY1, we went back to the field because we want to help the farmers in the end. We cultivated more or less 400 F2 individuals with my collaborator in the field. It's tough work. And when we use introgress PEPY1, the symptom is much less, and disease progress is much slow. But we cannot control the disease completely. But we can at least have double production just introgressing the PEPY1. So more recently, this is what we wanted to do in the end. We introgress both PEPY1 and PEPY2 in the same pepper plant, like we call say pyramiding. And for this study, we use, it's a little bit complicated, but for pepper genotype. Number 218 is susceptible control. G1 is a Capsicum chinense. It's kind of tolerant variety. And BA-PEP5 with PEP1, PG11 with PEP2. We check with the DNA marker. And we use two groups of begomoviruses. One is from Southeast Asia, Indonesia, PEP5CIB, PEP5CAB. Another one is from America, PHYBB, PEP-GMB. Those two are also famous begomovirus. Bipartite one, we prefer the infectious clones. And initially, we inoculate singly. So in case of BA-PEP5, so if there is a yellow circle, it means resistant. If there is a blue X, it means susceptible. So in case of BA-PEP5, it was resistant against Asian begomovirus, but not against American begomovirus. But in case of PG11, it showed resistance against both begomovirus. And we checked the disease symptom more detail and did a statistical analysis to support this conclusion. And we also checked the viral DNA accumulation. And BA-PEP5 nicely restricted viral DNA accumulation for Asian begomovirus, but not against American ones. And PG11 restricted nicely except for PEP-GMB. But I would conclude that PEP5-1 is useful in Asia, and PEP5-2 seems to be useful in both areas at least. Next, we want to do similar thing as a field. In the field, co-infection of different begomovirus is a problem. So we simulated this in the laboratory. Using the previous material, for example, we prepared a PG11 infected PEP5CIB, PG11 inoculated with PEP5CAB, and we did the grafting so that plant can be mixed infected with two viruses. We did this for Asian begomovirus and also American begomovirus combination with four pepper genotype. And the situation was completely different. Only BA-PEP5 showed nice resistance against mixed infection of Asian begomovirus, but not others. All others became susceptible. Also, viral DNA accumulation result showed the same conclusion. So to combat this situation, we did parameting. And in this study, we used PHYBB and PEP-GMB because this was the most highly pathogenic combination. We initially inoculate two viruses by agroinfiltration to number 218 susceptible pepper and did the grafting to different pepper genotype. So there was susceptible number 218, BA-PEP5, PG11, and F2 hetero has PEP-Y1 in homozygous state, PEP-Y2 in heterozygous state, because PEP-Y2 is a dominant gene. And F2 homo has both genes in homozygous state. So when we inoculate, the number 218 BA-PEP5 PG11 is completely susceptible, but F2 hetero has much less disease symptom. But the symptoms differ between plant to plant because it's F2 population. But in case of F2 homo, there was almost no symptom, even co-infected by two begomoviruses. And when we check the viral DNA accumulation, in F2 hetero, viral DNA accumulation differ from plant to plant. But when we introgress two genes in homozygous state, it was constantly very low amount of viral DNA accumulation. So it depends on the area. If your area has a severe problem, I recommend to introgress two genes in the homozygous state. But if your area has a mild problem, just RDR may be enough with heterozygous. So I would like to make a conclusion. In case of tomato, now we know 4 genes from TY1 to TY6. 4 genes cloned by Netherlands, Dutch team, Israel team, and Japanese team. In case of pepper, we have cloned 2 genes. I think there are not the third one yet. I hope someone clones the next one. And we are also working with other vegetables like cucurbit, cucumber, melon, and eggplant. We have cloned the first gene in cucumber. Interestingly, the first cucumber gene, CY1, is also RDR. And last month, we published a paper in eggplant. we named this gene EY1 because it's eggplant. It encodes a completely novel gene, exonuclease, SMN3. So there seem to be different mechanisms. So at least in each vegetables, we can apply the knowledge. But now we have more modern technology, like gene editing, EMS mutation. So maybe in future we can apply the knowledge across the plant species. And of course, we can import the knowledge to peppers. So most of our research and publication is available in our website or ResearchGate. So if you are interested, please visit our website. And for further detail, please contact me directly. And in the end, I would like to especially thank 3 students who conducted PEPY1 work, Mika and Nadia, and for PEPY2 Namiko work very hard. And also Erike Smarty from Indonesia. She is my senior who took PhD in same university in Kyoto in Japan. And several collaborators from Japanese university and my collaborator Taki. And also I would like to thank my funding from Japanese government. So this is all from me. Thank you very much.